Group 15 will be presenting an article on diarrhea-causing bacteria adapted to spread in hospitals. Scientists have discovered that Clostridium difficile, a gut-infecting bacteria, is evolving into two different species. One of the species has the ability to thrive in hospital environments. This species has the ability to survive even in the presence of disinfectants. Biological evolution of C. difficile began about 76,000 years ago, far before modern hospitals ever existed. Biological evolution is a genetic change that occurs in a population over the time span of many generations. A well-known example of this would be the evolution of physical structure of Neanderthals to humans. This includes phenotype or physical changes and genotype or genome changes. In the speciation of C. difficile, the genes involved in spore formation are changed and result in two distinct species. Evolution can happen through natural selection, and this is essentially when genes for a particular trait are more common in the offspring of an organism than they are in the offspring of another organism. For example, in a population of birds, birds with the hereditary trait of a large beak reproduce more than other birds with smaller beaks. Therefore, there will be more birds with larger beaks than birds with smaller beaks, and this is because birds with larger beaks are more favored by the environment. An experiment was performed to test the effectiveness of hospital disinfectants against C. difficile, and it was found that none of them was able to completely wipe out the bacteria in the biofilms in which the experiment was performed. This means that some of them died while the others survived. So in this case, those that survived had the adaptation, which is resistance to hospital disinfectant. So now we're going to go into the specifics about the experimental procedure. And an experiment was performed involving genome analysis to explore the characteristics of C. difficile. 906 strains of this bacteria were isolated from various sources and received by the Wellcome Sanger Institute. Phenylchloroform, which is a liquid-to-liquid isolation method, was used to extract DNA from all strains. Bacteria was cultured on blood agar plates mixed with yeast extract under anaerobic conditions for 2 days, and growth occurred within 16 hours. DNA of C. difficile was sequenced by implementing the Aleena high-sec machine. This sequenced DNA was then analyzed with phylogenetic, recombinant, functional, and pseudogenetic methods. C. difficile was grown using anaerobic incubation, centrifugation, serial dilutions, and this was repeated 3 times for clades A and B. Each spore was exposed to ethanol and then hydrogen peroxide to test which ones were resistant. C. difficile clade A thrive within the human body due to the ability to metabolize fructose and glucose, as these sugars are often consumed by humans. As C. difficile gains more energy, the production of spores increases. Thus, clade A was shown to create greater density of spores and is able to infect new hosts at a faster rate. Also, C. difficile, like many other bacteria, have gained antibiotic resistance through selection and horizontal gene transfer with other bacteria. With all those factors combined, C. difficile clade A has become a threat to hospitals across the world, with China having the highest percentage, followed by Europe and then the US. Overall, the study directs research into looking for new ways to control C. difficile, such as developing more efficient disinfectants or possibly analyzing and modifying human diets. On a worldwide scale, the study shows the need to genetically track bacteria to understand the threats they have on human healthcare and prepare future medicine for the rise of new species and mutations.